We're going to have another presentation from Rajiv Mayani, that is a researcher, programmer at ISI, too, in the Pegasus team, and he'll be talking about automated processing of phenotypic data submissions using Pegasus. Go ahead, Rajiv.
Hi, everyone. So I'll be talking about an automated workflow for curating phenotypic data. So this is a workflow that we developed for an NIMH-funded project, which manages a data repository at ISI. So just to give you a background about why this workflow was developed, is that NIMH basically funds many studies that carry out psychiatric research on psychiatric disorders like schizophrenia, bipolar disorder, and many others. And as part of these funded studies, the researchers usually collect biosamples such as blood, saliva, and phenotypic data, which is basically questionnaire kind of data. So if you are someone who's studying the effects of substance abuse on schizophrenia, the phenotypic data would usually be how often do you drink, how much do you drink, and questions like these. And so as part of these funded studies, NIMH mandates that all of this data be submitted to a centralized repository. So the biosample repository is managed at Rutgers. So blood samples, saliva samples are sent to them, and the phenotypic data is sent to ISI, which maintains the data repository. And all of this information is now then made available to other researchers. So a typical scenario for researchers would be that they would come up with a hypothesis like saying that I want to study the impacts of how schizophrenia is inherited on females of a particular race. And to prove or disprove this hypothesis, they would say, I would need to collect samples, blood samples, and ask some questions to approximately 5,000 participants. So the researchers in this case would come to the center and look at the available samples and data that's already accessible to them. And based on this, they could identify that there are 1,500 individuals who already meet the requirement. So now the researchers can request access to these samples and data directly from the center, and they can amend their grant to only request funding for collecting 3,500 blood samples from 3,500 participants. So, and again, as part of these studies, they would be required to submit back the biosamples and data from these 3,500 participants to the center. And so the AutoQC workflow was developed to validate the phenotypic data that these researchers submit to the center so that it can be harmonized in a single format and make it easy to search and access for other researchers. And so the workflow that we have developed is made accessible through a web interface. The researchers would basically create a zip file with all of their input files in a CSV file format. Each of their input files are associated with the data dictionaries that describe what type of data is included in them. So this would describe each column of the data, the type of data contained in that column, whether it's textual, numeric, the range of the data. So for example, age could be a minimum of from 0 to 120.
Rajiv, I think your screen sharing stopped.
Shows me I'm still sharing my screen. Can you see it now?
Yeah. You were at the next slide.
Sorry, can you repeat that?
Yes, I thought you were at the next slide while talking, so I was confused. Sorry.
Yeah, this is the next slide. Yeah. And so the data dictionary also contains information that describes the column so that researchers who are looking at this data for their own studies can make sense of the data contained in them. The one thing that we have done is that we have tried to standardize all of the data that the system requires, and at the same time allowing users to submit custom data that we wouldn't know how to standardize. So basic information such as the information of the individual, such as age, whether the person is alive or dead, the birth year, and these kinds of information has been standardized into a single template, so it facilitates easily searching for information across all studies from across disorders. And wherever we can't standardize the data, which is when researchers would come up with their own questionnaires with custom questions and custom rating mechanisms, they would submit the data dictionaries for their own data along with it. So the system can run these checks. So as part of the workflow, the first step we do is we check each of the input files against the data dictionaries, which could be either system defined or provided by the user, and followed by a bunch of advanced checks, which are custom coded and cannot be standardized. And so the workflow structure, as you see here, is as I described earlier. So if you see on the top, it's the set of basic QC checks, which is compares the data against the given data dictionary and validates it. And each of the boxes you see are the different types of standardized files that are defined. And on the right, the phenotypic validation are the custom defined files where the user provides both the data and the data dictionary. And in the second stage, we have custom checks where we do pedigree validation, which basically validates information about a family structure. So an example of this would be if an individual is identified as a father of another individual, then we verify that the gender of the individual is set to male and the age of the individual is greater than the individual identified as a child, and things like that. And then another set of advanced QC checks are an example of those would be where we validate the individual's information can be correlated with the information submitted to the biorepository at Rutgers. So say we say the blood sample ID of an individual exists and is valid and is associated with the correct individual. And now we just have some screenshots of the system. The system is basically broken down into 3 pages. The first page is that you see right now is just listing all of the previous runs or previous data submissions that the users have submitted, the disorder for which the data was being submitted, the file name, and whether the validation was successful or not is shown through color coding. The second page is basically the submission interface where the users would upload their zip file containing all of their input files, and they would specify the study ID, which is the NIMH associated study ID for which they are submitting the data, and the disorder of the associated data. And if the users would like to receive email notifications when the validation process has completed. And the final page here is the monitoring page where the users can track the progress of the workflow in real time and see the status of each step. So for each file, you would see whether it has passed the basic checks or not. And if they have failed, you can click on the log links to view the report and basically iterate over your input, make the corrections and resubmit it. The one thing that we have done is that we have made this process both real time and offline. So you do not have to stick on the page here to track the progress continued. You can start of your submission, go away and come back when you need to, or just wait for the email notification and come back and check the status of the correction of your validation run. And that's all I had.